Welcome to the How to Build a Microscope webinar series. Today we will be talking about transmitted light. I'm happy to introduce our presenters today. They will be Dr. Henry Haberle and Dr. Melissa Eberle. Henry is our application specialist at Thorlabs. Dr. Haberle received his PhD degree in neuroscience from University of California, San Francisco. He was a postdoctoral fellow at Stanford University School of Medicine before joining an imaging facility at University of New South Wales. He managed an imaging core at Columbia University. He will be doing the first part of our talk today. Melissa Eberle is our regional sales manager at Thorlabs Imaging Systems. Dr. Eberle received her BS in bioengineering from UC Santa Cruz and her PhD in bioengineering from UC Riverside. After her graduate work, she did a postdoctoral fellowship at Yale University before joining Thorlabs as our application specialist.
Thank you very much for that introduction, Pauline, and welcome everyone to part 2 of our How to Build a Microscope series, where we're going to be focusing on wide field imaging with transmitted light. This is the second part of our series, where we'll be building upon the fundamentals introduced during part 1 of this series. So in the first few slides, I am going to be reviewing the key elements of the first talk. So a microscope collects and magnifies light from the sample for imaging or detection. And the two key elements of modern microscopes are the objective and the tube lens. The objective collects light from the sample and relays it to the tube lens, which focuses that light through the eyepieces to the eye or to the camera or another detection apparatus. Resolution is usually limited by the numerical aperture of the objective. The numerical aperture of an objective is a measure of the degree of angle that that objective collects. A lower NA objective collects a narrow angle of light emitted from the sample and has less resolution and a longer depth of field. A high NA objective, by contrast, has higher resolution and a shallow depth of field. Continuing the review from talk 1, we're going to cover 2 of the major pathways in a microscope. Epi orientation, or the epifluorescence pathway, which illuminates the sample from the same side as the imaging detection module. Light is either reflected or generated within the sample. This is used for fluorescence and reflected light microscopy. This is in opposed to transorientation for transmitted light illumination. This is where you illuminate from the opposite side of the sample as the viewing apparatus. Light is transmitting through the sample. Resolution in this setup is usually limited by both the objective numerical aperture and the transmitted light numerical aperture. In today's talk, we are going to be talking about the fundamental elements of transmitted light path design, including going through Kohler Illumination and how that is an ideal design for a transmitted light path. We will talk about how to optimize using this light path for Brightfield imaging. We will then talk about the key element of transmitted light, which is the condenser, and the key specs of that element. We'll then talk about Brightfield illumination advantages and limitations, and then focus in the last part of the talk on several contrast enhancing techniques, including Darkfield, phase contrast, and differential interference contrast. So the fundamental goal of a transmitted light pathway is to light the sample. So how do you illuminate a dark sample? Well, the simplest way of illuminating a sample would be to put a light source directly behind the sample. This is advantageous in that a large fraction of light from the light source passes through the sample and is collected by the objective lens. In addition, many angles of light from the light source are passing through the sample to the objective lens, which, as we will see later, can improve your resolution. The disadvantages are many for this type of simple setup, though. The primary disadvantage is because the light source is optically close to the imaging plane of the objective, the light source will appear in the image. Furthermore, you have no control over this type of setup. You have no control over the angles of sample illumination. And you also have no control over the area of the sample that you're illuminating, or otherwise known as the field of illumination. These problems can be remedied partially by moving the light source away from the sample. So with the light source far from the optical imaging plane of the objective, the light will no longer appear in the image. And so now that is the advantage to this type of setup. However, this comes at a cost. With the light source being far away from the sample, now only a small fraction of the light rays from the light source make it through the sample and are collected by the objective lens. Furthermore, we still don't have any sort of control over this type of setup. This can be improved by introducing a collector lens between the light source and the sample. This relays an image of the light source to a point close to the sample. This collector lens, the primary advantage here is this collector lens will now direct a much higher proportion of the light from the light source through the sample into the objective lens, brightening the sample and increasing the efficiency of the transmitted light path. In addition, you have large angles of illumination of the sample in this type of setup. However, we've traded one disadvantage for another, in that now the relayed image of the light source is close to the sample and therefore close to the imaging plane of the objective, and therefore once again the light source appears in the image. This setup can be improved slightly by introducing our first movable element, the aperture iris, at the relayed image of the light source. This gives us a certain amount of control over the system, in that now if we close the iris down along the relayed image of the light source, we can decrease the angle of light rays that are striking the sample, and now we have control over the angles of sample illumination. To improve upon the fact that the light source is showing up in the image, we can move this entire structure away from the sample. So now the advantage here is, since the relayed image of the light source is far away from the imaging plane of the objective, we don't see it in the sample. But once again, we have the problem that only a fraction of the rays from the light source are making it through the sample and into the objective, and the pathway is inefficient and the sample appears dim. This can be improved by introducing another lens into the transmitted light pathway, the condenser lens. Now we are transmitting a large fraction of the light source to the sample, so the sample appears bright and the pathway is efficient. In addition, we still have large angles of illumination that we can control that go to the sample. Finally, we have the light source is relayed far away from the imaging plane of the objective, and therefore we don't see the light source in the sample. Our only disadvantage here is we still don't have any control of field illumination, or the area of the sample that we're illuminating. For that, we need one more iris called the field iris. Adding an additional iris close to the collector lens allows us to control the area of the sample that we are illuminating. This has many advantages and few disadvantages, and is therefore an ideal setup for transmitted light imaging. This setup is called Kohler Illumination and is named after August Kohler, who invented it. In this diagram, I'd like to walk you through some of the conjugate planes of the transmitted light path and the imaging light path to show you the advantages of this system. Starting with the sample and conjugate planes to the sample shown with the green light lines. You can see that starting from the left-hand side, the field iris is a focal point for the sample light rays, and therefore closing down the field iris will be conjugate to the sample plane and will illuminate a controlled area of the sample. The aperture iris is not in focus and does not affect the actual illuminated area of the sample. The sample is conjugate to the intermediate image plane where one could place a camera for imaging, and then it's relayed through the eyepiece lens and the lens of the eye and focuses on the retina, showing that the sample is actually showing up in the eye. Now I'd like to take you through the light source light rays of this system, which are delineated here in red. The light source, either being the light source itself or a diffuser element that one can place in the light path, is conjugate to the aperture iris. You can see the beams crossing there. And then the light is importantly collected by the condenser lens and then passes collimated straight through the sample. This illumination light then focuses on the objective backfocal plane and then is importantly not focused upon the retina of the eye, showing that the light source is not imaged by the eye. Now I'd like to take you through this diagram again, but now showing you some off-axis light. So this light is coming from the edge of the diffuser or the light source. You can see this light focuses through the edge of the aperture iris and then is collected by the condenser lens, and then while collimated is illuminating the sample at an angle. These are the light rays that are cut off when one closes down the aperture iris and therefore reduces the degree of angles that are illuminating the sample. Commercial light paths, such as the Thorlabs transmitted light path, are a derivative of Kohler Illumination light path, in that the diffuser is an additional element into the light path. So here, the diffuser is importantly conjugate to the aperture iris of the condenser or the pupil plane of the objective, rather than the light source itself, because essentially you could think of the light rays as emanating from the diffuser. In a properly set up system, the field stop of the transmitted light path will be conjugate to the sample and the imaging plane of the objective. Since samples are thick, condensers in a transmitted light path have the ability to focus up and down in order to illuminate the relevant parts of a sample. If you were looking through the eyepieces and adjust the field diaphragm of a transmitted light path, you will see the field diaphragm stop down the image in the microscope because the field diaphragm is conjugate to the image plane. This is as opposed to an aperture diaphragm, which you would not see focused at all in the sample. In fact, in order to see the aperture iris, one can use a trick by removing the final lens from the microscope or the eyepiece lens. Now with one lens missing, you can see from this ray diagram that the light source is now focusing onto the back of the retina. By removing this lens, you can now see the objective back focal plane, the aperture iris of the condenser, as well as the light source or the diffuser. If you are looking through a microscope without the eyepieces in, you will see this. You will see the ground glass diffuser in the image or the light source itself. And as you close down the aperture diaphragm, you will see this close down in your image. In addition, if the condenser numerical aperture is less than the objective numerical aperture, you will see the condenser in this image as well. When we place the eyepieces back in, you see the effect of this aperture iris on imaging. This is because when you close down the aperture iris, you are essentially changing the numerical aperture of the condenser by reducing it. This effect in the image plane is shown here. In the eyepiece view, you can see that the image starts brighter, but with less contrast. And as you stop down the aperture iris, you decrease the brightness and, more importantly, increase the contrast. So with Brightfield transmitted light imaging, you will see that you can have an optimization where you will either have the aperture iris open, which allows you to have high resolution, a shallow depth of field, or you can stop down the aperture diaphragm and have high contrast with a large depth of field. Now I'd like to go through a few key specifics of the condenser. So the condenser, the most important specification of a condenser is its maximum numerical aperture. Here I've shown you as an example our condenser from Thorlabs, the CSC 2001. It has a numerical aperture of 0.78. Two other key specifications for a condenser are the field of view that the condenser can illuminate. This is frequently listed as compatibility with certain objective magnifications. This is because high magnification objectives typically image a small field of view in the sample, and low magnification objectives illuminate image a larger field of view and therefore require a condenser that can illuminate a larger field of view. The final key specification for condensers is the working distance, which is the distance that the condenser focuses at, or the distance between the edge of the condenser and the focal point. There are two slightly less well-known specifications for condensers that home builders will need to know. The first is the pupil diameter of the condenser, which is the diameter of light that needs to enter the back aperture of the condenser in order to fully utilize the full numerical aperture of the condenser. And the second specification is the aperture stop location relative to the base of the condenser. This is so the aperture iris can be correctly conjugated to the rest of the elements of the transmitted light pathway. So on our example of the CSC 2001, it has a 2.5 millimeter field of view. And so in our marketing materials, you will see that that means its recommended objective magnification pairings are 10x through 100x. It has a working distance of 6.6 millimeters. Its pupil diameter is 33 millimeters, and the aperture stop location is about 27 millimeters from the dovetail. Using this type of transmitted light imaging is used for Brightfield imaging, which is the most well-known and widely used imaging tool in biology. The advantages are that it's easy to set up and inexpensive, and it's adjustable in that you can optimize contrast versus resolution. The disadvantages are that contrast between, or that trade-off between contrast and resolution, which is paired with another trade-off between depth of field and resolution. Furthermore, it's not ideal for thin, very transparent specimens, which can remain invisible even when the aperture iris is closed down. Therefore, very transparent specimens require some sort of additional labeling in order to see. The main applications are for fixed and labeled cells and tissue, or naturally high contrast tissues such as pollen. Finally, this is the imaging mode that's most frequently used in the medical settings to image stained biopsies. So in this first part of the talk, I've taken you through transmitted light path design, culminating in Kohler illumination, and how it is ideal transmitted light path design. We talked about how to optimize Brightfield illumination. Then we talked about key elements of the condenser, and then the Brightfield illumination advantages and limitations. For the last half of the talk, we are going to be talking about several contrast enhancing techniques that can improve upon Brightfield illumination. This includes Darkfield, phase contrast, and differential interference contrast. We'll start with Darkfield illumination. In Darkfield illumination, you place a Darkfield annulus within the condenser lens that's large enough to block light from being collected by the objective. That is, zero order or non-diffracted light escapes the side of the objective and is not collected by the objective. That's opposed to diffracted light that's scattered by the sample. A fraction of this light will be diffracted in the direction of the objective and collected by the objective and then the microscope for imaging. Therefore, these type of images have a dark background since none of the zero order light transmits to the image, with bright samples that have diffraction showing up bright against that dark background. This type of illumination is a simple method to increase contrast in transparent samples. Furthermore, these transparent samples are highlighted against a dark background. Advantages to this setup include that it's simple to implement in existing condensers and it's inexpensive. It highlights the outlines of transparent structures, and therefore you don't need to label the sample. The disadvantages include that it requires a lower numerical aperture of objective than the numerical aperture of the condenser, and therefore with that lower numerical aperture objective, some fine details will be obscured. Its main applications are in live cell or live animal imaging, especially for small transparent specimens like bacteria. Furthermore, outside the life sciences, this method is used to image transparent crystals that are made of mineral or chemical. In addition, it's used to image thin slices of non-transparent material, such as ceramics. A second contrast enhancing technique is called phase contrast. Here, a condenser annulus in the objective is paired with a phase ring in the objective. So the condenser annulus blocks low and high spatial frequencies of light and lets only a ring of light pass through the sample. This ring is then matched by a ring in the pupil plane of the objective that contains a plus or minus quarter wave plate and a neutral density filter. In this setup, zero order non-diffracted light passes directly through the sample and passes exclusively through the phase ring of the objective, where it's subjected to the phase plate and the neutral density filter. This is opposed to diffracted light, which once diffracted at the sample, a fraction of which is collected by the objective, most of this light passes through the other areas of the objective that are not subject to the phase plate or neutral density filter. So what does this do for you? Well, this allows you to help increase contrast. So in this diagram, I show you both phase and non-phase contrast setups and show you how the phase contrast system can increase contrast for transparent samples. So you have incident light to a sample plane. Then importantly, you have the diffracted light and the zero order background light. For thin biological structures, the scattered light is delayed by about a quarter wavelength relative to the undiffracted light. This quarter wavelength shift is not very well visible though, because it's much dimmer or smaller in amplitude relative to the background light for most transparent specimens. And a quarter wavelength shift does not lead to large amplitude changes in the image of the sample, and therefore the sample remains transparent. So This is in opposed to phase contrast objectives, which then delay that scattered light by an additional quarter wavelength, or I'm sorry, delay the background light by a quarter wavelength or advance that background light by a quarter wavelength. In addition, because of the neutral density filter, the background light is attenuated relative to the light scattered by the sample. Now the scattered light and the background light are either a half a wavelength apart from each other, leading to destructive interference, or are in phase again, leading to constructive interference. This will emphasize the sample relative to the background's illumination. Phase contrast samples, therefore, are highlighted against a gray background. The advantages is that this is simple to add to a transmitted light pathway. It highlights the outlines of samples, and therefore transparent samples do not require labels. Disadvantages to this setup is that it requires specialized objectives. These objectives with phase rings and neutral density filters are not ideal for other imaging modalities, including fluorescence, because they are not transmitting light efficiently. In addition, this type of contrast method generates a very distinctive halo around objects. Finally, the phase annuli in the objectives do reduce the resolution of the image slightly. The main applications to phase contrast imaging include live cell imaging, especially of thin scattered specimens where the halos around the specimens do not interfere with imaging. And for the last of the contrast enhancing techniques and the best of the contrast enhancing techniques for differential interference contrast, I'm going to hand this talk over to my colleague, Melissa Aberle.
Hello, everyone. Thank you, Henry, for that talk. Good afternoon, everyone. I'm Melissa Aberle, and I'm going to be discussing the last imaging contrast method called differential interference contrast, or DIC. So here are two images of HeLa cells. This one here on the left is phase contrast image showing the iconic halo effect for phase images that Henry just discussed. And over here is the DIC image of these cells. And an identifying feature of a DIC image is the three-dimensional appearance you see in this image. So this is because DIC imaging highlights the edges of objects. So the outline of the cells, as you can see here, as well as internal organelles actually stand out, not only from the background, but actually from uniform areas within the cells, which have the same brightness level as the background. Another feature for DIC images is that the contrast is directional, where one edge of an object, or here the cell, will appear brighter, and the other side of the cell will appear darker than the background. And this is as if the illumination is from the side. So what is a DIC image, and how is this edge contrast generated? So DIC is a polarization-based dual beam interferometric imaging method where changes to the light passing through the sample provides the image contrast. So here's an overview of the DIC microscope light path. So polarized light is split into two orthogonally polarized rays that are spatially displaced or sheared at the sample plane. The rays travel through the sample and actually interfere before observation, and the resulting interference pattern reflects the optical path differences experienced by each of these beams as they pass through the sample. So I will go step by step through each of these optical elements and describe how a DIC image is produced. So the first element is a polarizer. So transmitted light from the module, the WFA 1000 that Henry described earlier, will deliver collimated light to the polarizer up here through the polarizer, and the polarizer will convert the unpolarized collimated light into linearly polarized light at a 45-degree angle. The next optical component in the light path is a prism, the condenser prism. So polarized light now passes through a Wallston prism, and the prism splits the input beams into spatially separated orthogonally polarized rays of equal intensity. So these rays are often referred to as ordinary, or O in some of our pictures, and they propagate perpendicular to the optic axis. So they'll appear blue or as a line in our diagrams. And then the other is the extraordinary ray, which will be represented as an E, and they propagate parallel to the optic axis. So they'll appear red or as a dot in our diagrams. And so how does a Wollaston prism work? So this, a beam of light polarized at 45 degrees will hit this prism, and it'll actually hit the Wollaston prism, which is made of two wedges of different birefringent materials. So the first birefringent material will actually apply a phase delay between the E and the O rays. So you'll see there's actually a separation to the E and O ray as it goes through this first wedge. So it's either referred to as a phase delay or retardation between the E and the O. When it hits this wedge, there's actually a difference in refractive index, which causes refraction at the boundary and splits the light into two spatially separated orthogonally polarized rays. So it'll actually separate or shear these E and O waves. And then the light will pass through, these rays will pass through a second birefringent material that actually has an opposite optical axis. So then you'll actually start to see a delay of the E versus the O wave, and as those rays come out on the opposite side, you'll see that on the bottom part of the Wollaston prism, the E wave will actually be ahead of the O wave. And what that looks like in space is that what that phase delay actually looks like in space is here you can see in this diagram, it's actually a shift in the peak amplitudes. So there's actually, one of the waves actually slows down with respect to the other. And so you actually get this phase shift between the two peaks of the amplitudes. And that's what we're referring to when we're saying that one is actually ahead of the other. It's actually the peaks and where these amplitudes line up. So at the lower part of this prism, E is ahead of O, but on the upper part, here we can start to see that the O wave ray is actually ahead of the E wave, and that's responsible for how much of each of these birefringent materials the beam has to pass through. And so it actually creates, as you can see, with these kind of diagonal lines, a diagonal waveform as this beam propagates out of the Wollaston prism. As the beams propagate, these two orthogonal rays leave the Wollaston prism. They enter the condenser, and they are rendered parallel into the sample. So beams that propagate through the edge will enter the sample at an angle, and the rays that propagate through the center will enter horizontally. And then once they pass through the sample, they'll be collected by the objective. So these rays in this diagram are actually exaggerations of the sheared rays to show the separation. However, they're actually close together, ranging from around 1.6 micrometers to about 0.6 micrometers, depending on the condenser NA. So you're actually comparing beams that are very close together. The condenser prisms are matched to DIC compatible objectives, which are typically engraved with either an N1 or an N2. So here's the Thorlabs condenser, the CSC 2001, and then two options for a visible or an IR condenser prism. So the next optical element is a second Wollaston prism. So after the two sheared rays travel through the sample, they are collected by the objective and pass through another Wollaston prism that's placed conjugate to the condenser prism. So it's very important that these two prisms are matched and they're placed conjugate to each other. This second Wollaston prism actually has an equal opposite effect as the first Wollaston prism to the phase delay of these two, the E and the O beams. So as you can see in this image, the E and the O beams are delayed from one another. They actually pass through the opposite birefringent effect as the first prism, because now they're passing through the bottom part, and they will emerge linear and in phase. The same will happen through the center of the condenser, the prism. And so the light emerging out of this second Wollaston prism will be linear if there's no effect from the sample. However, if the sample does have changes in refractive index and or thickness, the two beams will experience differences in optical path lengths. So the optical path length is the product of the refractive index and the thickness of the object. So basically one of these rays will actually take longer to travel through the sample than the other, and thus this will be phase delay from one another. So here is an example again of what phase delay of these amplitudes would look like, where these red and blue arrows representing the peaks of these propagating waveforms. And if there's a phase delay and then this second Wollaston prism aligns them, elliptically polarized light will actually emerge out of the second Wollaston prism. And that is important because the last optical element for DIC is an analyzer, which is a polarizer that forms a cross pair with the initial polarizer. So here's the initial polarizer, and this is the analyzer, which forms the cross pair. So starting from the most simple form, as if there was no sample and no effect, light leaving that second Wollaston prism will be polarized linearly and will be completely blocked by this cross-paired analyzer. So, however, if there is some phase delay caused by the sample, then you'll actually get a varying degree of elliptically, circularly, or linearly polarized light that will then be allowed to pass through the analyzer. So then you'll actually be able to get signal on the other side and be able to detect an image. So the analyzer actually recombines these ordinary and extraordinary planes of the ray pair, and this allows for interference to occur. So if there is a phase difference, it's converted into amplitude due to constructive and destructive interference. So if the amplitudes line up, you'll actually see a reinforcement or a brightening of the signal. And if they're out of phase, then you'll actually get destructive interference or a decrease in the signal. And so basically what the analyzer does is the resulting differences in intensity, it'll show resulting differences in intensity when viewed in a wide field camera or the eyepieces. So it effectively converts differences in optical path length across the field of view into differences in intensity in an image. And so here's the WFA 3110 DIC analyzer that we have. You have a visible analyzer, an IR analyzer, and an empty slot for Brightfield illumination. So here's some example DIC images and movies. So you can see from this data that the edges are more prominent because there are large phase shifts between the two sheared beams. As one ray goes through the sample and the other goes through, say, the edge or the slide or other similar tissue. So here we have two cells dividing in time. Here we have a mouse retinal layer, 50 micrometers deep. We have cheek cells where the nuclei and edges of the cells are very distinct. You have very nice contrast, as well as a pollen grain where you can see the nice details of the edges of the pollen grain. So there's one more step to optimize a DIC microscope, and that is the introduction of a phase bias retardation into a DIC light path. And you do this via a compensator. And the reason we do this is, we'll take this left as an example. So this is a trapezoidal sample, and we're going to imagine that an E and O ray are passing through this sample. And so both, you can see, are slowed down in proportion to the change of the optical path length, which again is the product of the thickness and the refractive index of the sample. And the wavefronts you see here, the wavefront you see here represented is after the objective Wollaston prism. So the Wollaston prism will realign the E and the O rays, and the phase differences then can be observed between the two rays at the edges of the sample. So region A is the background, and so you see that there's no phase delay as it goes to the analyzer. It'll appear dark. Here at B, there is an edge, and so there is a phase difference for the length of the edge, and so you'll actually see an increase in intensity after the analyzer. At C location, however, both of the rays pass through the same amount and type of tissue, and so there's actually no net phase delay between the two, and so it'll actually look dark within the cell or in this trapezoidal sample. And then at D, you hit the next edge, which again has a phase difference along that edge, and thus an increase in intensity is observed. So, however, you can see that this image is very difficult to look at. It's a very dark background with only very light kind of blips where you have edges. And so the idea is to better be able to visualize DIC images is to add a bias retardation, to be able to pull this signal out of the background, or out of the background and off of that kind of base noise floor. And so how that works is you basically want to, a phase retardation is introduced. So here's our waveform after the Wollaston prism again. And the idea is to advance one of the rays in order to brighten up the background. So now phase offsets or phase shifts from the sample can appear both bright and dark from the background. So here in A, you see there is a phase shift, so you'll see a change in intensity, but you'll actually see a larger change in intensity at the edge of the sample. And then in C, again, you see a shift or retardation in the phase, but it's still going to be at a higher background level. And here in D, you can now start to see that an opposite effect from the side of the edge of D. Now you can see a decrease in intensity. So now you have more of a dynamic range to work with, and you can see brighter and darker areas away from this sort of gray background. And that's why it's important to add a compensator into the DIC light path. So there's two methods to generate this bias retardation. One is the Nemarsky method. And the Nemarsky method uses a Nemarsky modified prism, objective prism. So one of the edges is, so here's the modified prism. One of the edges is identical to the conventional Wollaston wedge, and it has the optical axis oriented parallel to the surface of the prism. And the second wedge of the prism, however, is modified so that the optical axis is oriented obliquely with respect to the flat surface of the prism. And so this Nemarsky modification causes the light rays actually to come to a focal point outside the body of the prism, which allows for greater flexibility when setting up the microscope, as now the prisms can be actively focused and you can actually put them in both places, in both prism locations. And how you then use it as a compensator to kind of select for that bias retardation is you actually slide this prism in and out, which is why it's referred to as a slider. And so you can slide it to a certain degree to sort of select the best image contrast. The second method is a Desceramont method, and the bias retardation occurs with the addition of a quarter wave plate between the polarizer and the condenser prism. And so how the Desceramont compensator works is you actually rotate the polarizer through plus or minus 45 degrees or a quarter wavelength without changing the orientation of the quarter wave plate. So this allows you to control the bias retardation incidented on the sample. And so here you can actually see what it looks like if you go through the plus 45 degrees and the minus 45 degrees. You can actually select the direction of the phase shift gradient. So you can actually select the shear angle. So one will either delay the E versus the O ray, or the opposite, where the O ray will actually be delayed versus the E ray, and you can actually see the shadowing shift in the opposite direction. So up here you have the dark edges on the top and then the light edges on the bottom of the image, and in this image the bright edges are on the top and the dark edges are on the bottom. And so this allows you to actually fine-tune to better select the contrast for your specific sample. Here is kind of images for what going through that polarizer rotation would look like. Here's a very small percentage of change in wavelength. So useful ranges for bias retardation are often between like 1/13 and a quarter wavelength. So here you can see as you rotate through the polarizer and those different angles, you can start to see the loss of this really nice edge contrast, and you can start to see it kind of starts to blend in with the background. And then so samples with very large optical gradients can often have even greater bias retardations up to a full wavelength. So this is one of those things where you kind of do it with your sample on there and rotate the polarizer to kind of fit your sample. So for DIC, it's kind of the gold standard for transmitted light imaging contrast enhancement. The advantages are you can see fine details in thin, transparent specimens clearly visible, as you saw in some of the data I showed. There's no labeling needed. It works well, actually, with up to about 100 micrometer thick samples, so that's nice for tissue work. Cells appear to be shaded as if illuminated from the side, allowing for this really nice contrast method, making not only cell walls, but internal organelles and structures really obvious with nice contrast. Another advantage is it can actually be paired with other imaging modalities, such as fluorescence, as you see here in this kidney cell slide. Disadvantages are it's the most expensive transmitted light method that we talked about today, mainly because those optical components have to be well matched and well manufactured, so they're pretty expensive. It does not work well with very thick samples, and it doesn't work well on fixed samples. And that's because fixing samples actually changes the index of refraction and thickness, and so you don't get as nice contrast. So some of the main applications are electrophysiology, where you can see slices in cells over time, as well as cell and specimen development, as we showed in that cell video, where you can actually do DIC imaging over periods of time. So today we discussed the transillumination pathway, including several contrast enhancement techniques such as Darkfield, phase, and DIC. There is also epi-illumination pathways for fluorescence or reflection microscopy, which we will discuss in more detail in future talks. So after today's talk, we've now covered the basics of microscope design, including the transmission illumination pathway. The next talk will cover how to build microscopes yourself using Thorlab's parts. And then future talks will cover multiphoton imaging and other advanced imaging techniques. So thank you for listening today. Here's some contact email and phone number if you have further questions or want to get in contact with us. And I will now pass the question and answer section over to Pauline to moderate.
Thank you, Melissa. I'm bringing back Henry as well. Thank you for the talk. So I'm going to ask some questions that some people have posted. So first question is, what is the best way to illuminate through a fiber? Do you need Kohler illumination when you don't have a filament? Is a collimating lens enough?
So I'm assuming the fiber is fairly thick. I mean, yeah, you can definitely image fibers with Brightfield imaging, and if you stop down the aperture diaphragm with that really long, you can achieve a really long depth of field. So yes, you could use it with, you could definitely image a fiber without one of the, other contrast enhancing techniques we have, just with straight Kohler illumination and a properly adjusted condenser.
Okay. And I think, so I'm going to ask the next question, which has to do with the NA of the condenser. Somebody asked, if you set the NA of the condenser to less than the NA of the objective, will the effective NA of the objective be lowered, hence equal to the NA of the condenser?
Yeah, so for transmitted light imaging, the numerical aperture of the condenser and therefore the transmitted light path definitely does affect the resolution of the final image. So yes, when you stop down the aperture iris, and or if you have a condenser with a low numerical aperture, yes, you will have a lower resolution transmitted light image.
Mm-hmm. So I guess going back to the first question, if the fiber and the collimating lens that they pick produces collimated light that is less than the entrance pupil of the condenser, then effectively they would decrease the NA. Is that correct?
Yes, absolutely. Yeah. If you underfill the condenser pupil, then you will be effectively reducing the numerical aperture of the condenser.
Yeah. Sounds great. Next one, the question that we have is basically somebody's asking, to image small minerals and cells, would Darkfield be the best contrast method?
Which contrast method to use is a frequent subject of debate. Darkfield is a great method. I think it can really come down to the details of your particular sample. And what the setup on your particular microscope or the microscopes you have access to is in terms of your financial constraints. It's hard. There's no one way to answer that question.
Okay. Next question that I basically have is for Melissa. So we have dot contrast on our website along with DIC. Can you talk about the difference between the two and why you would select one over the other?
I guess it would really depend on your sample, so, and it, DIC will, kind of what you're interested in looking at. So DIC will give you, better kind of edge and internal organelles of the cell. If you're looking at cells, kind of highlighting the structure, where phase or dot be a little more like phase, where you might not necessarily get the kind of full structural effect out of that image. You'll be able to visualize it, but you might not necessarily be able to see the contrast, the difference between, say, an internal part of the cell versus that organelle. So it kind of depends on what you're interested in looking at, and what you're trying to track over time. Like if you're just trying to track like the edge, of a cell growing, then maybe that, dot could work. If you're interested in something going on like within a cell or within an organelle, like a C. elegans and an organism, then it might be better to use DIC, because then you can actually get kind of better contrast of smaller, organelles kind of moving through the system.
That sounds great. I have another question about DIC. So somebody's asking if they can do reflection DIC for semiconductor inspection. Would there be a difference between a tungsten halogen lamp versus a mercury arc lamp?
You can do reflection DIC. I don't know if there's a difference between the two light sources and how they perform. I don't know. Yeah, unfortunately, I don't know enough about the difference of those light sources. Whether you would get it basically you basically just need kind of a polarized beam. So you can, both of those you can collimate and then polarize. So I actually don't know whether one would be more advantageous over the other. Henry, do you know, or?
No, I have no idea.
Yeah.
We have another question here. Question is, what if the sample is moving? How do you illuminate it uniformly?
I guess I'll take that one. A sample, well, in a properly Kohler illuminated system, you should have pretty even illumination across your field of view. So even if a sample is moving, you should have even illumination, so long as it's staying within within the field of view of the objective and the condenser.
Okay. I have a follow-up question here. Basically, could you explain the aspects of Kohler illumination in reflection mode, and would it differ from those in transmitted mode?
Honestly, I don't know, because if you were to reflect light, Kohler illumination is, in terms of the transmitted light pathways, is for transmitted light. I'm a little bit confused by the question. I guess you could refer in terms of the Kohler pathway in terms of the rest of the imaging side of the pathway, but I'm not familiar with that.
I can take the question. Yeah, so
Sure.
in epi mode, so the epi fluorescence modules typically also have, an aperture and a field stop. So basically, yes, they're using the same type of concepts into ensuring basically that you have even illumination. So yeah, it's and because you're going through the same optics, meaning in this case you're going through the objective, back and forth, then the NAs are basically well matched.
Yeah, and if since you're using the objective in order to focus on the sample, you know that you're at the focal point.
Yep, exactly. 10. Another question that we have here, I guess, can we go back one slide, just going over what the future sessions will be about?
Yeah, give me one sec. There you go.
Okay, so the question is basically what kind of future sessions we'll be holding in the imaging and microscopy series. So yes, our next one will be about DIY microscopy, and then we'll do one about on fluorescence imaging, and that's where we'll probably cover most of the topics for epi, and then multiphoton and advanced imaging techniques. Okay. I think that's it. That's all the questions. Again, thank you very much, Henry and Melissa, for presenting today. And for everyone who's joined the talk, I hope you have a great day.